Hello students. Yesterday we have completed the chapter Living World and we started with the biological classification where I have given you the introduction about the different classification systems. Okay, so we have gone through about the five kingdom classification system. Now today we are going to discuss about the different kingdoms. Okay, so one by one let's start with the kingdom Monera. Kingdom Monera. Okay, so kingdom Monera belongs to which organisms? Let me take you through the introduction of the kingdom Monera. Kingdom Monera is the kingdom which belongs to the prokaryotic organisms. Okay, so this is the kingdom which is made for the prokaryotic organisms. These organisms are most primitive one. They are the most primitive organisms. They are most primitive organisms. The organisms in the kingdom Monera they have different characteristic features. They have the different characteristics of kingdom Monera. Okay, so if we talk about the kingdom Monera, as I have already told you in the classification system, that kingdom Monera, it contains mainly which organisms? It contains the bacteria group called the Eubacteriae. Okay, Eubacteriae and Archaebacteria. So these two types of the bacteria, Eubacteriae and Archaebacteria, they comes under the kingdom Monera. Now different characteristic features, like what characters are present in whole as a kingdom Monera. Okay, now number one, they are most primitive organisms. They have a prokaryotic organization. These organisms under the kingdom Monera, they have prokaryotic organization. That means they are made up of by prokaryotic cell. They have the prokaryotic cell. And these prokaryotic cells, you know that I have already taken you through about this concept that prokaryotic cells, they do not have well developed nucleus and nuclear envelope. So such organisms coming under the kingdom Monera, they do not have a well developed nucleus or nuclear envelope. Okay, also the organisms coming under the kingdom Monera, these organisms do not have membrane bounded cellular organelles. Okay, so these organisms in the kingdom Monera, they do not also have the membrane bounded cellular organelles. Fine, so continue with the characterizations. Next, except one organelle, they have only one organelle present in the kingdom Monera, which is membraneless. That is membraneless organelle, and that organelle is known as ribosome. And especially in prokaryotes, the type of the ribosome which is present in them, it is called 70S type of ribosome. That is the 70S type of ribosome. Remember this thing. Apart from that, I told you in last lecture as well, the bacterial cell inside their cytoplasm, of the cell, they do not have any other cellular organelle apart from the ribosome. But in the cytoplasm, these bacteria, they have food storage bodies. So the food is going to be stored in the form of glycogen bodies. The food is going to be stored in the form of phosphate granules, and those phosphate granules, they are known as volutin granules. Fine. And they can also store the food in the form of fat bodies. Okay, that can be also stored in them. And also they have presence of enzymes, various enzymes in the cytoplasm. Okay, so the cytoplasm is filled with these food storage. Okay, now if we talk about more about the kingdom Monera, as I told you, the kingdom Monera is mainly represented by bacteria. Bacteria is the sole organism of this kingdom. Okay, so let me take you through the detailed structure of the bacterial cell, the features, the characteristics which are present in the bacterial cell. That feature, that characters, they represent the whole kingdom Monera. Okay, so let me take you through the structure of the bacterial cell, where you will come to know about what characteristic features are there in the bacteria as well as in the kingdom Monera. Okay, so let's discuss about the bacterial cell. Fine. See, let me give the structure of bacterial cell, typical bacterial cell. Okay, so the outermost covering of the cell, it is made up of by the envelope system, and this envelope system has different layers, like the layer which is I am going to make, it is the outermost layer of the bacteria, which is present on their outer surface. Okay, like this. And this outermost layer, it is known as glycocalyx layer. Okay, the outermost layer, which is a smooth layer, granular layer, and sticky layers as well, this is known as the glycocalyx layer. Now inside the glycocalyx layer, there is another layer which is known as cell wall. It is known as cell wall. Fine. Now inside the cell wall, there is another layer. Inside the cell wall, the another layer is there, the layer which I am going to form here. Now this layer inside the cell wall, it's called cell membrane. But this cell membrane that I will explain you later on one by one, inside what is the composition of glycocalyx, what is the composition of cell wall, what is the composition of cell membrane. But structurally you remember as of now, this cell membrane which is inside the cell wall, it shows enfoldings at the different locations where it goes infold at the central position, as well as the cell membrane shows infoldings at the corners. Okay, these structures which are going infold of the cell membrane, the structure which is infold in the center, it is called central mesosome. Those enfolding structures are known as mesosomes, and the central position it's called central mesosome, and there is a mesosome at peripheral location is known as peripheral mesosome. It is the peripheral mesosome. Okay, so these are the two different types of the mesosome, that is the modification of the cell membrane. Remember this. Now if we talk about externally on the body of the bacterial cell, what other structures they may have the presence. Externally in their body there arises the some structures like this, which are short as well as the long. Some are in the short length and some are in the long lengths. Some are the short lengths, some are the long lengths, like this. Like this. These hair-like structures, they are known as, these hair-like structures, they are known as pillars. They are known as pillars. The longer one, they are known as the pillars, as well as the shorter ones, they are also called the fibrils. Fibrils. They are known as fibrils. Structurally, they are same. There is no difference in structurally, but functionally, there is a little bit difference, which I will explain you later on. Okay. So these are the some structures present on the body of the bacteria externally. Also, externally on the body of the bacteria, they also have the presence of certain granular-like structure bodies nearby to the cell membrane, from where a plate-like structure is being developed like this, and it forms a hook-like structure. Then a long elongated structure is being formed with that. This is known as flagella. It is known as flagellum. Single, it is known as flagellum. Okay, but if there are the more than two or one, then it's called flagella. Similarly, they are going to be formed like this. A long elongated structure, it is known as the flagella. This flagella arises, it has three parts. This flagella has three parts. These granular like structures, they are known as basal bodies. They are the centriolar bodies from where the flagella is starting to form. Okay, that is known as the flagellar body. From there, a plate like fibril structure is going to be formed. They are known as fibrils. Then plate like structure is being formed, then hook like structure is being formed, a little bit curved structure, that's the hook, and then long elongated part, it is called a filament. Okay, so this whole thing together, it is known as flagella or flagellum. Singular, it's flagellum. When it's more than one, then it's called the flagella as a plural. Okay, so those are the structures present on their external body, and these flagellar structures, they make the bacteria motile, so they helps the bacterial motility, movement of the bacteria. So they helps the movement of the bacteria. So bacteria does movement from one place to another place with the help of these flagellar body, so with the help of these flagellums. Okay, so these are structures present on the external body of the bacteria. Okay, remember this. Now if we talk about inside, in this cytoplasmic region of the bacterial cell, this is the cytoplasmic region of the bacterial cell. As I told you, in the bacterial cytoplasm, they do not have any membrane bound cell organelle present, but they have a membrane less organelle only, that is the ribosome. That is the ribosome. So these ribosomes are like scatteredly formed inside the cytoplasmic region. Some of the ribosomes get associated nearby to the mesosome structures like this. Like this. Some of the ribosomes get formed nearby the mesosome. These ribosomes, like structure nearby the mesosome, they function as respiratory enzymes. They helps in the respiration, enzymatic respiration process. They helps in that, so they are called respiratory enzymes. Now these granular bodies present in the cytoplasm, these are the ribosomes. 70S type, that you know that. Okay, apart from that, these bacterial cytoplasm, inside the cytoplasm, in the central region, they have a presence of highly coiled double-stranded thick material, genetic material, which is here in the central position. It goes coagulated at the central position. Double-stranded DNA. This is a double-stranded DNA. This forms as a genetic material, which is the genetic material of the bacteria, which is double-stranded DNA. But this DNA, it doesn't contain outermost covering like a nucleus. तो न्यूक्लियस में क्या होता है बेटा, जैसे अगर आप यूकेरियोटिक सेल में देखेंगे तो वहां पे प्रॉपर न्यूक्लियस होता है, न्यूक्लियस की कवरिंग होती है, फिर कवरिंग के अंदर ये डीएनए होता है, आरएनए होता है. बट यहाँ प्रोकैरियोट्स में क्या है बेटा, यहाँ पे कोई न्यूक्लियर एनवेलप नहीं है. There is no envelope system. This whole genomic DNA, that genetic material, it is completely naked. And it is naked but double-stranded DNA. It contains double-stranded DNA, plus it has the combination of some proteins called non-histone proteins. Okay, so eukaryotes have histone proteins. Those prokaryotes like bacteria have non-histone proteins. Those non-histone proteins are called polyamines. So in this region, this is the genetic material, genomic DNA of the bacteria, which contains the non-histone proteins as well, and it has the presence of RNA also. This whole structure gets coagulated in the central position, and this is known as nucleoid. This whole structure is known as nucleoid. Remember this point. In bacterial cell, they do not have well-developed nucleus. They have undeveloped nuclear structure which contains only DNA and non-histone proteins, which is naked, completely naked, and this nucleoid is naked, and that naked structure is known as nucleoid. Okay, remember this point. This nucleoid is also called genophore. It is another name having genophore. And this nucleoid is also known as incipient nucleus. It is also known as incipient nucleus. So they have a different names of the same structure, have a different name. Either you called it nucleoid, either you called it genophore, either you called it incipient nucleus. So basically ये जो एरिया न्यूक्लियर एरिया होता है ये बिल्कुल नेक्ड होता है, इसमें डीएनए डबल स्ट्रैंडेड हाईली कॉइल्ड होता है विद द हेल्प ऑफ दीज नॉन स्टोन प्रोटीन. इसी पूरे रीजन को हम बोलते हैं न्यूक्लियोइड, जीनोफोर और इंसिपिएंट न्यूक्लियस. ठीक है, सो रिमेंबर दिस पॉइंट, दिस इज इन द साइटोप्लाज्मिक रीजन. Apart from that, inside the cytoplasmic region of some bacteria, not all bacteria, in some bacteria, certain bacteria, apart from this genomic DNA, the main DNA of the cell, apart from this, some bacteria, they also have the presence of small circular double-stranded DNA. At certain places, they have presence of small circular type of double-stranded DNA, and this DNA is known as plasmid DNA. It is known as plasmid DNA. This DNA is different. This DNA is different from this genomic DNA. Okay, remember this. So it is not present in all the bacteria. It can be seen only in certain bacteria. And those bacteria which contains the plasmid DNA, they become special type of the bacteria rather than this typical bacteria. Okay, that we'll discuss later. Apart from that, other structures present in the cytoplasmic region of the bacterial cell. What are those? The bacterial cell also has the presence of food storage. Food is going to be stored in the form of different bodies, granular bodies, like they stores the food in the form of glycogen granules. So food is stored in the form of glycogen granules. Then the same food is going to be stored in the form of another type of food bodies. They are called volutin granules. देखो बेटा, volutin granules जो होते हैं, they are the phosphate granules. They are phosphate granules. यानी bacteria stores the food in the form of phosphate granules. Those phosphate granules are called the volutin granules. Remember. Also, bacteria, they store the food in the form of fat granules. They can also store the food in the fat globules. So also fat bodies are being stored in the bacterial cytoplasm. Apart from that, these bacteria have a numerous type of enzymes present in the cytoplasm, and those enzymes, they are helpful for the various metabolic reactions. These are the various enzymes. Okay, so these are the basic characteristic features of the bacterial cell, that what are the characteristics present in the bacterial cell. This whole bacterial cell represents the whole kingdom Monera. So now you can get a good overview about what type of the organisms or characteristic features they have under the kingdom Monera. So these are the characteristic features they have presence in under the kingdom Monera. Okay, so now there is these layers, outer layers, inner layers. What are these characteristic features? One by one we have to discuss about these characteristic features to make more understanding their composition. Like glycocalyx layer is made up of what, cell wall में क्या होता है, cell membrane में क्या होता है, और अंदर इनका function क्या होता है, that we need to go in the next. Okay, so that I will explain you in detail in the next lecture. Thank you.